Sometimes you have the opportunity to present or to introduce researchers, really, really good researchers. So, and in this case, when you present someone you admire, it's quite better. So let's introduce, it is my pleasure to introduce you Andrea Malandrino, who is a tenure track Serra Hunter Fellow in Universidad Politécnica of Catalonia, where he is part of the Material Science and Engineering department in the group of Biomechanics and Tissue Engineering group. Dr. Malandrino actually have the master in mechanical engineering and did his PhD in biomedical engineering from Universidad Politécnica of Catalonia and the Institute for Bioengineering of Catalonia. And I have to say that during that time, during past 1.5 years, I have been in the asked PhD candidate, myself, that actually have the patience to teach, and I'm really grateful for that. After that, he got a Marie Curie fellowship and went to MIT for Roger Kahn, Professor Roger Kahn. And after that, he joined the PRBB in the, let me check, don't forget, European Molecular Biology Laboratory. Basically, his interests are focused more in, of course, biomechanics, mechanobiology of the dynamic of the extracellular matrix. Basically, he's a huge, let's say, toys, let's say microfluidic chambers, basically to actually model how diseases work, to study the biomechanics, and also if he can find patterns to organogenesis. This part of the talk actually combined two fields that are not combined. Javier said at the beginning that clinician and pharmaceutical doesn't like us, that's one part. And many times, experiment and modeling does not combine. It's really weird, and we need each other. So in this case, Dr. Malandrino actually combined both fields. That is something really nice. So Andrea, the point is yours.
Thank you, and this is extremely nice introduction, so thank you for that. I don't have to say anything else, I guess. Thank you to remember all my trajectory. That's a little bit curved. So I'm going to talk about extracellular matrices and mechanical plasticity. This is the outline. I'll introduce the topic. And then I'll put some example of studies we have been doing over the last 7 or 8 years, I think. And also the last part is something I'm doing right now in Germany, as I am a visiting researcher in Erlangen, Friedrich Alexander University. If I have time for that, we'll see. Okay. Extracellular matrix, you probably don't need much introduction. Is what I don't know how to define it properly, but it's stuff produced by cells that in the end is all that is not cell and is extracellular. So the name speaks by itself. And there are several types, but it's basically what we are built of together with cells. So it's pretty important stuff. Makes up 70% of our dry body mass. People tend to consider a passive material because if you take it out and kill off the cells, you can test it as it was a normal material. It's like a passive, but when it's in the body, the cells are actually remodeling actively, and it's not a passive material. So it's active in this sense, so it's a living material. I am actually I'm a mechanical engineer, but I work for a material science department. So this is interesting because as a material, this is a living material. So a material that is not static, is evolving. So it's not prone to the same tools that we have in classic material science to be studied. So remodeling, homogeneity, etc. How the cells interact with this material in a number of ways, of course, biochemically, etc. But we are interested in how they interact mechanically. So all the field of mechanosensing, I'm going to talk mostly about fibrous ECM, so which is like collagen or fibrin, which is the material of, for example, clots in our when we have a wound, or collagen, which is the main protein of our body, extracellular protein. So all these are fibrous material, and cells interact with these fibers, and I will show an example of that, and mechanically. And we can measure pretty many things mechanically with this. It's important to note and to highlight that is a living material, and also it's very inhomogeneous, so it's not really a continuum. We cannot treat it as a continuum unless we do very smart homogenization procedure, which not always work, because sometimes we have to consider that this homogeneity is important. Anyway, this is an engineering student, mostly audience, so you probably have seen that. This is a stress-strain curve, the textbook stuff of a metal. It just serves me because plasticity can mean many things. There is cell plasticity, synaptic plasticity, even psychological plasticity. We are talking about mechanical plasticity. So, and mechanical plasticity is you can load the material up to a point in the elastic regime, and then you remove the load, the material goes back, and it's a conservative process. But you can have an inelastic effect if you surpass this load and enter the plastic regime. Sometimes you fracture before that. You enter the plastic regime, you remove the load, and then you have a residual. In this case, it showed a residual strain. So you go back with the elastic route, but your material is permanently deformed. This is what I'm talking. And it's important. I will show you some. We are doing many analogies with this plasticity in our fibrous ECM. And we are also looking at residual stress. It's an important feature of plastic material. Residual stress is again, with an example of metals. I didn't find a better analogy than a katana. This is a steel, Japanese sword that is differentially cooled in the nucleus and in the part that is you know, and because of this residual stress that are generated during the different cooling process, you have martensite, more carbon and more hard, where you need hardness, and then you have a more ductile. Because of this residual, you have this nice curved shape. So it's a way, residual stress, also to sculpt a material. And that's important, because when we go to tissue, this is one of my favorite papers of the last 10 years. And it's not my paper. Sometimes you have favorite papers that are not yours. You say, why I didn't do this? These people took a slab of collagen and then precisely placed cells with a very sophisticated coating, precisely placed cells in this collagen, and then let them do their mechanical work. Mainly it was a mechanical stress they applied. And then this cell deformed the material, and they were doing a computational model and predict many shapes, just depending on where you place the cells, and they exactly predict how the material would deform and would stay deformed. So this is an example for my own work, which I will show you later. These are capillary endothelial cells that form vascular network. And then at some point, when I was at MIT, I decided to study the mechanical interaction between the ECM, which is the blue part. It's a fibrin gel, ECM-like. It's not the real ECM, a surrogate. But also the collagen slab they were using are a surrogate of ECM. I killed the cell, so you see there is this GIF, the cell disappeared, and the material was completely sculpted and remodeled, and there is no way that would come back to the initial random orientation of fiber they have. So we started to think about, hey, this is kind of a plastic process, very different from what happens in metals, of course. So the analogy is a little bit poor, but we move this way. So this is going to be this is one of my favorite movies that we took, and I keep playing it because, and sometimes, I use this method of careful staring before analyzing anything. A careful stare at this movie so much time that I know exactly how each pixel move, I think. But I want you to, the colors are probably not the best, but I'm sharing the screen from Zoom, so, I have the presentation here. This is the most digital twin thing that I've done today, and I will talk about today. So I want you to notice that there are protrusion of the cells. This is the first thing I wanted to notice, that this seems important. I wanted to notice that the whole network, this is a collagen hydrogel with fibers, the whole network is hugely moving, so there is deformation. But I also wanted you to notice that there are this accumulation of fiber close to the cell, which we call stiffening. It's a kind of armor that the cell is building. And this video is hours, like, let's say, tens of minutes. So this is a fast process, a fast remodeling process that has happened as, it was an afternoon. I went to the microscope, and in 2 hours, I took this movie. So it was very fast, and I say, wow, this is fast. And the cells are remodeling and doing this change in material very fast. So we started to studying it. Okay. Maybe I have to stop this. This is so. Okay. So first thing, we concentrated to this stiffening. There is two kinds of stiffening in this system. There is one stiffening by concentration. The cell drag fiber close to them, and there are more fibers in some region and less fibers far away. So what we did is to use optical tweezer in a collaborative project. This is when you embed these beads. They have a different refractive index from the media, and you can move them by pointing a very highly focused laser beam, so you know the force you're applying. So we disperse these beads around the cells and then move them, and then we know the distance of each bead from the cell. So what we discovered is that the closer the beads were to the cell, the stiffer was the material. You can already have guessed that because there are more fibers. There is this armor that the cell is building. The beads that are far away are less stiff. Okay. But then there is another stiffening process. Each of these fibers and the whole fibrous collagen thing is stiffening because of entropic elasticity, bending, and buckling of fibers. So you have a material that if you take this collagen and you start to stretch, you have this curve, force displacement. So you have a material that is less stiff, and as fiber align on the directional load, you have a very stiff material. So there is another kind of stiffness, the non-linearity of the fiber mechanical behavior. So what we did is to call a group of modelers from the Netherlands, and they built this lattice model where each fiber has a nonlinear stiffening term that replicate this, and then put a cell, contract the cell, and then we were giving them the stiffness that we calculate from the optical tweezer experiment. And they were extracting the stress corresponding to that stiffness. And what we discovered with this, we needed a model. That the only way to analyze those data is to build this computational model, because we only knew this stiffness from optical tweezer and the deformation you can observe in the microscope, but to get to the stress, we needed a computational model. So they built a model, and that was the only way to analyze this data. And what we discovered is that the material presents a property that is called long range for stress propagation. If you have an elastic material and you have a force, this force propagates to the cube of the distance. If you have this kind of fibrous material, it propagates farther away. So a cell that is here, another cell that is here, applying a force can reach a cell far away in a very efficient way, much more than if it was an elastic material. So this was a property we discovered for this kind of materials. For some reason, I cannot remove these things, so they were reference, but I will put because it looks like this thing, I did it all by myself, but there is a reference here. Where? But I cannot even see my pointer. Anyway, maybe I'll upload the presentation and there are the reference. This is a study that we published in PNAS in 2018. Okay. I think it's because of the screen sharing and stuff. I'm already happy that people can hear me and I can use my computer to play the videos.
Just give me a couple of...
I think you have to do ESC and then stop share. Okay.
So here, what do you put in?
Play for the presentation, yes.
Okay.
Okay.
See then. And
no, it's this.
you need to swap the screens.
Ah, okay.
Yeah. So I don't know how you can do.
Swap the screens.
Because this screen should be actually over there. So.
But I am.
If it's PowerPoint, I know how to do it, but not here.
I have no idea. I think. Okay, I'll just cite the reference and then share my presentation. Okay. Good. Then we start to research the biological or the cell features that will produce more or less of this stiffening around the cells of this fiber. And then we target protrusion, of course, because cells have this little arm, can be lamellipodia, can be filopodia, of actin that polymerize and make the cells move around and send out this protrusion and use actively this fiber. So if we have a drug that blocks protrusion, like CK66, SMIF2 or latrunculin, then we had no remodeling and no plasticity or stiffening, permanent change in the material configuration, let's say. But if we use a drug like GM6001 is a drug that acts on the degradation, so MMP, matrix metalloproteinase, so not directly on the protrusion of the cells, that didn't have any effect. You always can see this remodeled feature, like I'm pointing here with the arrow. So we developed a theory, and of course here again we needed a model to see how far away we were. The theory was, okay, the cells send out the protrusion, grab on the fibers, and the fibers are all cross-linked between them, so the cells is applying a force to the fiber that exceed the stability of the crosslink until the crosslink fails, and the cell can drag the fibers, and then this process continues on and on until more fibers are dragged closely to the cells. That was our idea. So mainly depending on the unbinding of fibers from the rest of the collagen or the fibrin network. So how we test this? We first drag the cells with cytochalasin D and applying no forces. So a cell that is under cytochalasin D will not send out protrusion, will just apply no force. Then we wash away this, and the cells start to doing this, the loading phase. And then when we can use, this is called PIV, particle image velocimetry, to infer the deformation that the cell is applying to the material. And then we can unload again wash away the cells, put a detergent, the cell is completely killed. Why we do this? Because in the first part, there would be elastic process and inelastic remodeling derived process that we can quantify. Whereas in the last step, just killing the cell and relaxing the material, we just have elastic, at least. That's what our intuition tells us. So if we compare these two, we may tell something about this inelastic or plastic deformation. And that's what we did. There is this guy, so this study was published in 2019 in PLOS. Again, there is the reference down there, but it's not visible. What we did is to try to simulate this. For that, we needed a discrete model, not a finite element model was not good enough. A discrete model where we can model each fiber or random fibers, cross links, and then try to simulate, this load from Philopodia. You should see the video here. This dragging of fibers. And then see what we can understand with this model. We understood the model told us that there is a 4-step process, which is the one that I explained. And it's mainly based on unbinding. The cells drag on the fibers, unbind the fiber because it breaks the crosslink, continue again, and put all the fibers close, as I already told. And this is what the model predicted. Of course, we said, OK, but is this an elastic process? The answer is already evident from these experiments. Because when you remove the cells, the network, the fibrin network, in this case, the fibrous ECM, does not go to the initial configuration. Things have happened. It's a new material, plastically deformed. You may agree with the word plastic or not, but inelastically deformed. From the PIV, we calculated displacement, and then we had this kind of curve. So no force, then we have 0 strain, and then all this process from no force of loading is taking place, and then you reach some deformation, and then you unload, and you only have this. So all of this is residual strain, OK? And some elasticity as well. So this is pure elasticity. This is because this has happened in less than 1 hour. Of course, there is elastic process going on there, but there are also plastic processes. So at least we could separate these two words, OK? And then the screen model was a collaborator from Yale that did it. And then he said, a continuum model would be nice. You have experience with finite element. OK, I'll do it. And then I used the Van Toff elastoplastic and viscoelastic model. Probably you've seen this in textbook. You have a plastic element, you have a viscous element, you have elasticity in both. And also we introduced damage. Damage has been nicely introduced by Javier. So it's just a degradation of the elastic modulus of both the elastoplastic and the viscoelastic element by some parameter. Why damage? Because we are thinking it's an unbinding process. You actually disconnect fiber from the network. So the cells is damaging locally the network. So we said, because the model was not able to predict anything if we didn't include damage. So damage has to go away. This is all not strong evidence, but it's the best we could do. And then we simulate with finite element a cell contracting and apply the same loading that we are applying in experiments, no force, loading, unloading. And then when we have damage, we could reach things that were more similar to the experimental range. And if we have just a viscoplastic model with the properties that we plug in for this case, we didn't reach this. Of course, there is a huge standard deviation. The experiments are like this. You don't have it. You cannot fit perfectly the curves, but seems to be the best we could. So what we have learned, I think I can skip that because it's just a first summary, but I think that it's very, I already talked about all this property, long range damage, non-elasticity, stiffening, but we learned a lot from this single cell experiment. And then we said, okay, let's move, we learned the most fundamental, the mechanistic aspect of this. Okay, and then I say, let's move on more realistic stuff, not a cell in an ECM, but group of cells that are together in the ECM. And then I was in this lab in Boston where they were doing this vasculogenesis. Vasculogenesis is the formation of a network of capillaries. You always hear about angiogenesis, because angiogenesis is what happens throughout all our life, remodeling of our vasculature. Vasculogenesis only happens in the embryo. But it's a very interesting process because you see this video, but it's not actually very visible, but there would be better video. We could do it on a chip. We had a microfluidic chip. We put endothelial cells and other cell types. I will talk about the other cell types later. And then we have this process. The cells are isolated, but a very high density at the beginning. And in a matter of 48 hours, they connect and they form capillaries. I mean, you are mostly engineers, and this is a biological process, but the fact that we can do it on a chip and we can image to this high resolution, you can already see the potential it has. You can use this to do many things, including perfuse. I don't know if you see the beads, but once this network has formed, you can perfuse cells or beads and look how they travel the inside of the vasculature, apart from doing these very nice movies. And you can study a lot of things. My obsession was the ECM. So I said, okay, intracellularly, intraluminal, inside the capillary, I don't care what's happening. I'm only caring about the extracellular. How can I study this? And then, first of all, I stared carefully stared at the experiments and looked at this video many times and see, look, these cells are connecting. They are remodeling of these fibers. What are these fibers doing? And then also a higher resolution. And of course, there was remodeling, plastic deformation. Okay, and then I went to the cell mechanics community and say, how do you study cell matrix interaction in a plastic environment? They say, no plastic. Forget about plastic. We don't do plastic. Okay, what do you do then? We put the cells on top of a polyacrylamide gel, and we have beads. And then these beads, of course, there are no springs. These are just schematics. But these beads are embedded. The material is elastic. And then you make the cell load the material. And by just looking at how the beads moved and knowing the properties of the polyacrylamide, the stiffness, you get to the force the cell is applying. Cell mechanics is a field that is largely based on this technique. Okay, cool. I said, I'll do the same with my vascular network. I took my capillaries, and I just throw the beads inside and look at how the cell relax by some drug that will make the cell fluffy and relaxed, and then try to infer the force. It didn't work. Didn't work because you really need a lot of beads for doing that. And then we had this fantastic idea. Roger had this fantastic idea. Say, why don't you use the fibers themselves? Label the fibrin. You know how to label the fibers, the fibrin or the collagen. And why don't you use the signal of the fibers that is more dense than the signal of the beads that you can put in there and are also toxic for cells? And then I did it, and it worked beautifully. I haven't published this yet. Well, yes, I did, but what I did is to take some stages in the vasculogenesis process and kill the cells, and then look at this material relaxation, deformation, and running PIV, so we can know how the cells are locally deforming. It's not very good resolution, but this is what we get. And then try to understand what's happening. Day 0 to day 7, these are capillaries that are forming. You can see there is a peak at day 3, and then the cells stabilize in these capillaries, and there are no more peaks. And we also can see there are strains, because we had to stop there. Because all this thing of plasticity, you cannot calculate, you cannot talk about stress if you know the material is plastic. The material is not the same from day 0 to day 7. The material has changed, it has stiffened, and a lot of things have happened. So we stopped to strain. And then we could see that if we took cells that didn't connect, endothelial cells that were too far away, and they didn't do capillaries, these peaks didn't stop. I have more data that I didn't put here. So what we concluded is that the material stress and strain the ECM around until they have found a purpose, which is forming a capillary network. After that, things are pretty much stable in terms of mechanics. That was our main conclusion. But all these things went into an application, which is we were already knowing and seeing from many experiments that if we just put endothelial cells alone, the cells will form the network, and in 3-4 days they will die. But if we combine with other cells, like stromal cells, fibroblasts, endothelials and fibroblasts, they will stay, this chip, this vasculogenesis on a chip will stay alive for 2-3 weeks. You know, tubes that are very stable, and you can watch them for 2-3 weeks. But why is that? Nobody knew. And we did a lot of things that went into publication in 2023. Among them, my mechanical ECM studies. And what we understood, we did some nano-indentation with AFM. What we understood is when you have this case, JCC, justacrine is when you put fibroblasts and endothelial in the same channel. PCC is when you put them in different channels, but they are communicating chemically, not mechanically. So what we saw is that when you put them together in the same channel, the elastic modulus of the cell plus ECM increases a lot. And that correlates very well with the stability of the network. So the fibroblast's main role is to mechanically control the formation of capillaries. We managed to publish this in a very nice journal, and that's our current theory. So mechanics of the ECM is important for making this capillary network, although you won't think about it because the vasculature main role is to provide nutrients by diffusion or by other chemical processes, and you don't think about mechanical role of capillaries, but we found this, and it was very important. Let me check the time, because I think I have the capacity of over-explain stuff and lose time. We still have 20 minutes, right? 50 minutes? Okay. I'll show another case that I'm doing right now in Germany. It's related, again, to plasticity, my main obsession in the last 10 years. And these are called microtissues. Microtissues are chips where we have these pillars, PDMS. You put cells. The cells self-organize. You don't have to do much. They will do it because they cannot adhere to the walls. They will self-organize, and they will move these pillars. And then you apply analytical force balance, and you can calculate by just looking at how the pillars have moved to get closer. You can know the force that the microtissue applies. So it's a way to measure the static global force. It's very nice. So when they showed me, and I was there in Germany, I said, I want to do this. I want to calculate force, and actually I want to calculate residual force. So I want to do plasticity with this. Ben, that is my supervisor in Germany, told me, I don't know about that, but of course you can do it. I don't know if it's useful, but of course you can do it. Yes, why not? So how do they self-organize? The cells just come together, and in a matter of 1 week, they proliferate, and the only thing they can do is to attach to this pillar, and they apply forces, and you can measure this force. You can do it with muscle cells, and that's very cool, for example, and you can apply some calcium loading, and then you can make the cells contract and measure the contractile force, for example. That's one of the million of applications. I did it with fibroblasts. and then kill the cells and see, but not the collagen, and see if there is any residual stress. That was the idea. But also, to do that, I used a more local measurements, which are these green beads. The red is the cells, the blue is the collagen, the ECM, and the green are polyacrylamide beads. These are elastic beads. Why I wanted to use that? Because once I kill the cells, but the collagen stays there, if there are residual local stress, the beads would stay deformed. A bead would, if it's round here, if there is no force from cells, and is ellipsoidal, it has an aspect ratio higher than 1, if there is deformation. So I used these beads and the readings from the pillars, both combined. So first thing, we did like a dirty experiment. Let's see if we relax, we go all the way to 0. No, we didn't. So there are residual static force on this. How do we relax? We put, this is important perhaps, we put a detergent, it's called Triton X. This will make a lot of holes in the membrane of the cells until the cells is not able to do anything, it's completely killed. And not only that, we leave it for 2 days. So to make sure, outside of the incubator, so to make sure the cells is dead, it's dead. And then we were sure that we have residual stress. So there is something in the collagen structure that has changed, and the material doesn't go all the way back to 0 force. And to me, this is very interesting because it has to do with the property of collagen. Also confirmed by the beads. The beads didn't go back to their round shape. They stayed a little bit ellipsoidal. The round shape will be 1, the aspect ratio 1. But after relaxation, which is the second group here, there's still 1 point something. So the beads are staying deformed. They are entrapped in the collagen network. But what is mechanistically that explains this? Why is this residual stress happening? That's our question. So we started to do, people didn't believe me in the lab, and they say, okay, take the cells and apply Triton X, the bead don't go all the way back to spherical. Leave it 2 days more. Make sure they are all killed. They don't go back to round shape. Disconnect them with a tweezer. They made me do that. And you have to do that as a scientist because otherwise people won't believe you. Disconnect it from the pillars because there may be some residual force balance between the pillars and the cells. Disconnect one. The beads were still not round. Okay, get rid of collagen. Apply collagenase and dissolve collagen. The beads were round now. So it's the collagen the responsible of this residual stress, the ECM. So this experiment was a pain in the neck, but very useful. And then I called my friend in Yale, Michael, and I said, hey, we've done this study of the protrusion together, blah, 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 and, but now I'm seeing these things. Can you do a model for that? Can you do a fiber model? Can we explain from which molecular feature of the collagen this residual stress depends? Or which molecular feature is responsible for this residual stress? And he was very excited, of course. This is the same video than before. I don't know, you play it again. And then what he told me is, my model is telling me that the only way we can have residual stress is that fiber rebond. So it's not like before unbinding of fibers and dragging fiber, but the fibers are disconnected, and for some cellular process, they rebind, and a new fiber configuration is created. And then I said, I've never seen that. And then he sent me this paper and say, in fibrin it happens, in collagen nobody knows. And what are you using? I'm using collagen. Okay, then we are screwed. So in fibrin, these people demonstrated that mechanical loading at the network scale causes spontaneous formation of new bonds. It's like the fibers have uncovered cross-links that have not been used, and then during all this mess, the fiber can rebind together. Is this happening also in collagen? Hopefully, otherwise I haven't explained my data. So, yeah, so our hypothesis were new bonds forming under force leads to residual stress buildup, and also dynamic protrusion from cells must be related to this, because without cell loading, all this rebinding cannot happen. So how do we test this? We test this, we thought about using collagen glycation. Glycation is like a, you know, when you cook onions and they caramelize, Maillard reaction, you can do that, of course, not cooking the collagen, but you can do that by putting sugar on your collagen solution, and then you have a more sticky, let's say, collagen, where there should be more cross-links. The problem of that is that you are varying also stiffness and plasticity. So that was not very clean. And also to target protrusion, we can use drug, to test the second hypothesis. Long story short, glycation didn't do anything on the residual stress and on the total force, but the lamellipodial drug, RAC1, inhibition of RAC1, actually reduced both the total force, but didn't do much on the residual stress. So these are very new results I wanted to share, but we are still we're still doing experiments with a lot of cytoskeletal drugs, and we found some interesting effect of TGF-beta that would increase recruiting and stuff. But I don't have them ready, so I said, for the moment, we are seeing that our second hypothesis may be more valid. But we don't know how we can vary the cross-linking density of the collagen without altering its stiffness or its plastic behavior, ultimate strength or whatever. So we're still a little bit stuck, but it's getting somewhere. So this is a summary, but I think I already highlighted all this. If I have 5 minutes more, I'll just, yeah, I do. Okay, I'll just talk about non-fibrous ECM, because right now I'll talk about collagen, fibrin, blah, blah, blah. But I'm also running experiments on non-fibrous ECM. Why is non-fibrous ECM of interest? Because of all the bioprinting and tissue engineering world. Although there are very good recent advances, but bioprinting doesn't work very well. So printing a bioink is material and cells, an ECM-like material and cells. And you print it with a 3D printer for tissue engineering. You probably have heard of this, right? Fibrous biopolymers are not good for bioprinting. It's not very easy to print collagen or fibers or fibrin. Although we would love to, because we know them very well, and we know that their best cells is very happy in a fibrous biopolymer. So we have to use non-fibrous, like alginate. So these are videos that I took when I arrived in Germany. The first one is just alginate plus gelatin, and as you see, after 7 days, and as you see, the cells don't do much, don't spread, and so I said, you know, I'm not going to work with this. I always work with nice material, collagen, fibrin. The cells are happy there. Why are we working with this? But then I heard there was something going on in the lab, which is altering the pre-cross-linking state, and I will explain what that is, of alginate and gelatin, and having a material that is more colloidal, less viscous and impenetrable, like alginate, and where cells can spread more easily. And that has to do with plasticity. So I said, okay, this is my project. I jump in. Plasticity, I mean, non-fibrous material, but it's completely different from the plasticity I've been talking about now. It's more, so people know that cells mechano-adapt to viscoplasticity and they respond better, but we think, and I will provide evidence for that, that is more related to cells protrusion breaking intermolecular bonds of alginate gelatin. What we're using here is this mess here, which is an alginate, not a simple alginate, but I oxidize dihaldehyde alginate to have more functional groups. And we are mixing it with gelatin and have this that is called ADA gel. In Erlangen, where I am now working, there is a group of Professor Boccacini that has been using this, this material for a decade now. They have discovered that, so this material, you mix alginate and gelatin, very simply explained, and then you cross-link it by submerging in a bath of calcium. Calcium ions will make the material cross-linked, so polymerized, and be like stable mechanically. And then you can do that with cells inside, and that's the way you do it. But you can do something that is pre-cross-link. Basically, putting calcium ions, but not in a bath, but just spraying. We're now thinking about using a milk shaker, but this is a new story that I don't want to, because I still have to try. What we did is to put a nebulizer, that we bought it on Amazon for people that have asthmatic, but it was very powerful. And then you can put a calcium chloride solution, and while the other gel is preparing, is stirring, you spray this calcium. Like that, you obtain a material that has a slightly better printability. This is an extrusion stability study where you put the material in a bioprinter and you see what is happening. And this is the other gel control, and this is the pre-cross-link, but with more calcium ions and with less. We call this low pre-cross-link and high pre-cross-link. All the material are going to be bathed at the end. They all going to be finally cross-linked with calcium chloride. But these two are pre-cross-linked with the aerosol, okay? And then what we did is to microindentation to see if we are varying the stiffness. And it looks like over 20 days, the stiffness is very similar. So we are having a material that is more pastry colloidal, better for cell spreading, but has the same stiffness. So what is changing? And then Ben, my supervisor there, told me it's probably plastic things that is changing. I say, okay, bingo. Okay, so then what we did, how we test this material that, for some plastic properties. We thought about this. It's so simple that it looks ridiculous, but it works very nice. You put the material and you put an acupuncture needle, because acupuncture needles are very small, the smallest we can find, down to 100 micron diameter, I think, 150, I don't remember. I will show you now. And then you put the thing in a very high precision scale, and then by Newton laws, you are recording the force as you penetrate this material. Why is this giving it? So look at these movies. You penetrate an alginate gelatin that has not been pre-cross-linked. You have a high bending of the surface, and then, boom, you enter in. You penetrate this that have been pre-cross-linked, 40 and 60, low and high, sorry for the text, and you see that it enters very easily. Intermolecular bonds are not holding this, penetration force. We started to discuss what this is. We think it's related to ultimate strength. It is relevant for cells, yes, because cells live in the liquid phase. So yes, we are bending the surface, and we cannot measure the penetration force. I will show you why. The penetration force will be some slope of this, but as you can see, it's very hard. You have many layers of material that continues breaking in the highly pre-cross-linked. So what we measured, and it was the cleanest output we had, is these peaks. And you can see we are around 15, and here we are around 2. There is a 10, 1 order of magnitude factors in this. We call it puncture force. And this is something that has been already measured by other groups. So it's not, we didn't invent anything. but was never used for the alginate. And then we put this puncture force, and then we changed the needle diameter. We changed the acupuncture needles in 3 different diameters. This would be the one more relevant for cells. This is 700 micrometers. It's pretty big. And then you can see all the control have at least 10 times higher puncture force for all the diameters. And there is not much difference, and that's a problem. I will show you why. There's not much difference for the pre-cross link. So then we say, OK, puncture force gives a material that can be broken by, more easily. Intermolecular bonds can be broken more easily. So that's the reason maybe protrusion of cells. And cells can spread more easily in a pre-cross link material. And then we did a lot of, we already knew that was the case, but we repeated a lot of, this would be like how it looks like in reflectance microscopy, the material. So you see it's very patchy and disorganized and colloidal, but the cells love it and they spread in the pre-cross link much more than the control. Proliferation is enhanced. So cells are spreading, proliferating better in the pre-cross link material. And they are migrating. We did a lot of migration study, persistence, mean displacement. Cells move, spread, and proliferate as pre-cross linking is higher. So we tried to publish a paper. We're trying still to publish a paper. I don't know if I should say that, but I don't think any of you is a reviewer of this, where we basically say puncture force is a predictor of cell biological behavior in this kind of materials. And this, of course, will involve causation and is not blah, 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 Karl Popper. And we are not saying mechanistically why. We are proposing is because the cells can send out protrusion more easily and break intermolecular bond. But of course, we are not proposing a hard mechanistic behavior. So reviewers already replied a couple of times complaining this, you don't propose a mechanistic. But if I could speak to them, I mean, there is, in cell mechanics, everyone says cell stiffness correlates with cell biological behavior. And now if you take all together, all this data, it's a truth, it's a ground truth. But no one has said why mechanistically it is that. No one knows. So as Karl Popper said, there is no amount of data you can possibly collect to have a rigorous proof. The best thing we can do is to put some evidence in favor of something. That's what I would like to say to the reviewer. And since you are all young researchers, you have to face this. People will always ask for, what's the mechanism there? Hey, can you spend $20,000 of drugs and prove that and go to nature? Yes, I can, but I will not prove the mechanistic. Anyway, stop that. But our main problem here is that puncture force correlates to cell biological behavior, but the two pre-cross linking cases are very close. So we don't have 3 points here, effectively. We have 2 points. And that's also the problem of this study. So I will have to do more experiments. But we are very confident this is what's happening. And that's very interesting. So thank you. If you have questions, I'm here. If I didn't go OK, fine.